as we'll see in a second, when you measure a field's reflectance from a satellite, the area that's being measured for the satellite is very, it's averaged over large areas. So for example, in this case, every time the satellite sees a 250 meter by 250 meter plot, it gives it one value. That's called the resolution. Now on a drone, you could get the resolution down to a centimeter. So I mean, every centimeter you'd have a value. With a satellite, every 250 meters you have a value. So the drone-based remote sensing applications is very, is much more detailed, but doesn't mean it's better. If you're going to take the NDVI measurements from an entire, say, island or a continent, yeah, satellite is going to be better. You're not going to be able to fly a drone over that efficiently, right? But if you want to take your NDVI measurements over a plot of land, a small plot of land, a drone would be ideal, okay? So we're going to look at this satellite here on the right called the Moderate Resolution Imaging Spectroradiometer, also known as MODIS. We're going to look at examples of how MODIS measures NDVI. In this case, we're going to use Jamaica as a test study, okay? And that I actually did some research on this a couple years ago. It was published in the International Journal of Climatology, looking at how NDVI varies over Jamaica in different times of the year. This image on the top left is just a map of how much NDVI changes from the early rainfall season, say in May, to the midsummer, say in July. You can see this variation. What this map is showing is that in St. Elizabeth, there's a lot of NDVI reduction along the southern area of St. Elizabeth. And from my understanding, that is what's called the breadbasket of Jamaica, a lot of agricultural activity. And the point of this research was showing that, hey, in the midsummer, the NDVI, or the photosynthetic activity of this breadbasket area, decreases as you have a decrease in midsummer rainfall, okay? So we're going to look at this website. Here's an image of it on the right. It's called NASA Giovanni, and what it is, it's a web portal. So it hosts all kinds of NASA data, and we can access it, and we can plot it, we can map it, and we can use it. So what we're going to do is access the MODIS data that I just described, and we're going to look and see how MODIS measures NDVI over Jamaica, and look at the variability in NDVI over Jamaica, okay? So if you want to follow along with me, I'll give you a second to sketch down this. Oh, you know what? I'll put it in the chat. I'll put it in the chat, Control copy, and then you can, if you want, you can follow along there to that link. So I posted that if you want to open your browser. If not, if you just want to pay attention, that's fine as well. Okay, so I'm going to go, now I'm going to find my NASA Giovanni. One second, let me just set, I'm going to share a new screen. Okay, excellent. Where is this? Hold on. You would think after all this time of using Zoom, I'd be a lot more immersed. Okay, hold on a second. Meeting controls, where are you? Here we are. New share. Oh, I got to stop my share first. Okay. Now, here we go. New share. Screen 1. Share. Oh, great. Okay. So now everyone should see this web portal. Okay, so now this is what I get when I open the NASA Giovanni web portal. It's going to prompt you to log in, or you can continue with limited access, right? So that means you have to register, which is super simple. It requires an email and a password and like where you live. They just want to keep track of who uses it, right? So I've already registered. Registration is free. It takes maybe a minute. So I can go to log in. I'm just walking you through every step in this process because let's see, log in. Oh, here we go. Okay. So I'm just going to log in with my username. And my password. Okay, and I should also note, since this is being recorded, don't feel like you have to stress or get down all these steps. So keep it super simple, but you should also be able to access the recording and follow along in the future if you want to see more detail of what I did and how to replicate it, okay? But I'll keep it very simple. Okay, so I've logged in. Here's what the portal looks like. Fairly simple, okay? Now, on the left, you have all these different variables. To make it easy, since we know what we want to look at vegetation, let's click on the measurements triangle. I click on measurements, you have all these things that are measured that you can look at from NASA technology. I'm going to scroll down to vegetation. I see it right there. I'm clicking on it. See that 6 next to vegetation? That means we have 6 options to choose from. So I've clicked on it. I'm going to scroll up, and here we are. If I want to look at vegetation, NASA Giovanni, I can look at the greenness factor. Don't really know what that is, but I can guess what it means. Leaf area index, we're not going to look at that. EVI, that's a different vegetation index, but we're not going to look at that. NDVI, there we go. Another NDVI, another EVI. So what's the difference between these NDVIs? Well, 2 different types of MODIS satellites, Terra, Aqua. I wouldn't be concerned with that. I would just pick one or the other at this point. We're going to get monthly values. So we're going to get the average NDVI per month. Okay. And here's spatial resolution. And your data begins on February of 2000. And it goes up to the present, all the way up to, looks like last week. All right. Now, one might think, oh, I wish there was daily data. I wish I could get the vegetation of where my area every day. Well, the reason you can't do that is because clouds can obscure satellite vegetation monitoring. So if it's cloudy over your area, the satellite's not going to pick up any information. So daily data can be useless at that point, right? So what NASA does is say, hey, look, we're going to take all the information over 16 days and average it. We're going to call that, they call it a granule, okay? So that's why you don't have daily data, because of the potential cloud contamination, okay? So you take these roughly 2 granules and you average them into a month. That's what we're going to look at. So now we've determined we want NDVI. I'm going to click on one. So far, so good, right? All we've done is made 2 clicks, clicked on vegetation, clicked on NDVI. And now you go to the top, it's going to say, oh, well, how do you want to look at it? When do you want to look at it from? And where do you want to look at it? Okay. So right now, let's just look at the NDVI over Jamaica for last summer. Okay, so we're going to look at a time averaged map. I click on this, here are all my options. I'm going to look at a time averaged map. How do I know that? Because I want to look at a map, it's Jamaica, and I know it's time average because I said summer, that implies 3 months. But I'm actually just going to look at July. Only because when I run NASA Giovanni, it has to download a lot of data and make calculations in the background. So just to simplify it, I'm going to look at 1 month, the average of 1 month. Okay, let's just say July of 2021. Time average map. When's it start? 2021, July. When's it end? I think I can do the same month. If it gives me an error, we'll do 2 months. Okay. Okay, and now where is it? Well, you can enter the coordinates if you know them. If you don't, like I don't, you can click on this draw a bounding box on the map. So I click on that, a map comes up, and I can use my controls here to identify the region of interest. So I'm just going to use my mouse. I'm scrolling, make sure that hand is highlighted. I can pan it over. I want to scroll into Jamaica. And this usually takes a second to clear up, but there we go. Let's just do that. I might actually, okay, that's good. And now I'm going to click on the top box up here above the hand. That's my drawing box. I'm going to draw the area by clicking that and then holding my mouse down. I just clicked, dragged, and released. And now that entire area is highlighted. I can X out of this. And I know that's good because now look at my select region box. It's automatically filled in the geographic coordinates, okay? So I'm good to go. I've got a time average map. I know my month. I know where it is. I'm going to come down here and push plot data. Okay. I hope this doesn't take very long. It's crunching numbers in the background. That's why I only did 1 month and not 3. If I wasn't timed for a presentation, I'd probably do 3, you know? And here we go. All right, we've got some quick output. This is the average, I should say, the average NDVI for the month of July of 2021 in Jamaica. Here is our scale. Boop, 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 boop. You can see it goes up to, oh, look, it goes up to 99. But if you look at our units, it's times 10 to the minus 2. So it's actually 0.99. Okay, so basically it goes up to 1 and from 0.1. All right. So here's what I like to do. This kind of looks all the same to me. These values are so concentrated up in this upper region. I like this 0.61 to maybe 1s, right? So what I'm going to do, I'm going to adjust my color scale so that all the greens kind of range now from brown to green. I'll show you how I'm going to do that. You just go to options, click on options, and you go click on options again. And now I can change my data range. Okay, so let's have the minimum be 0.5 and let's have the maximum be 0.9. Let's just see what that looks like. Okay, I'm not going to change my color scales, but there's a ton to choose from if you wanted to. I'm not going to smooth it, but you can make it smooth if you wanted to. Scale it. Okay, now I'm just going to push replot. Let's see what happens. Sometimes I have to do this 2 or 3 times to get exactly what I want to see. Okay, and I'll explain to you how we can use this information and why or how it might be important. Now, do I get more variability? Yeah, I do. Look, okay, look. So if I change the color scale, I can see, oh, okay, let's change it a little. Actually, I'm gonna zoom in a little bit. Yeah, just zoomed in with my mouse, okay? So now I'm going to say, let's change this a little bit. Let's make these, how about 0.6? Let's have 0.6 to 0.85. All I'm trying to do is to increase my range of colors on the map so I can interpret it a little bit better. So options, 0.6 to 0.85. Now I'm hoping I get a better variation in color across the map. Let's see what it looks like. Was I right? Okay, I'm happy. I can tell I'm going to be happier with this. Look, now, just by adjusting the color range, color scale, whatever, I can now make interpretations more easily. I can clearly see, whoa, the vegetation index is much lower in southern Jamaica. Here, it's much higher. Here, it's much higher too. Now, a couple of things I want to mention right away. You'll notice, there are squares here. That looks kind of gross. Well, the square represents what's called the spatial resolution of the data. That means the satellite product is averaging all those 250 meter pixels into one, I think it was 0.05 degree resolution, so a 20th of a degree. Basically, that means that these squares are the average of a bunch of smaller values inside of it, all right? Second thing I want to mention, do not make the mistake of assuming, oh, just because there's low NDVI means the vegetation index or means the chlorophyll is low, okay? What do we know about this area right here? Well, this is Kingston, right? What do we know about Kingston? It's urbanized, a lot of land, a lot of concrete, a lot of buildings. Of course, that is not going to show up well with a vegetation index because it's going to say it's all zero, right? So this you can say is urbanized area, and I would not expect a high NDVI there. So therefore, I will not claim that, oh, the vegetation in Kingston is really poor. No. It's just there's very little vegetation relative to these other areas, right? Now, I do not have an expertise about this region. This is that southern St. Elizabeth region. Is this urbanized, or is this real reduction, not reduction, but real low level of NDVI? Can someone chime in? Can someone open their mic who's familiar with that area and tell me, is this a real vegetation index, or is this contaminated by an urban area?
Yeah, Teddy, that's a real index.
Okay, so there's a difference between the urbanization here relative to the urbanization there. So my point showing this is,
Yes, that area is one of the areas that basically suffers from long-term drought.
Yeah, that's what I thought. The southern St. Elizabeth region, correct?
Yes.
Yeah, so that's that map that we showed, or I showed in the earlier slide. So that's what I thought. I just didn't want to make the bold claim if others had greater expertise. So yeah, like Adrian's saying, that is the area that definitely experienced a lot of drought, and the farmers have a lot of coping mechanisms. They do different things in the midsummer relative to the late early summer for their cropping strategies. So one thing we can do now is say, hey, look, let's compare this area from 2021 to say July of 2020. Can we say that the vegetation was weaker or stronger in 2021 versus 2020? Well, let's check it out. So go back to data selection right here. And now I'm just going to change my data range from 2021 to 2020. Okay, and I'm just going to go to plot data. And you can see now I have a second map coming up. All the information of the first one is saved right here. I've got my plots. I could click on plot. It would show that map we just looked at. Now we change the data input. We're getting a second plot. And now I'm going to readjust my color scale so it's the same way as before, so we can make a good comparison. So options, like we did before, I believe the minimum was 0.6 and the maximum was 0.85. Okay, replot. And zoom in a little bit. Here's my 2020. And here's my 2021. Again, 2021. And here is 2020. All right. Now, you might be asking, oh, this is great information, but how can I use this for a presentation if I have to deliver this information to any stakeholders or extension officers or whatever? Well, what you can do, see where it says download? You can download this image as an image or Google Earth very easily. Just go to downloads. I'm clicking downloads. I have PNG, which is what's called, I think, a portable network graphic. It's just an image, right? So if I click on PNG, oh, that was quick. All right. Click on PNG. Let's see if I open this. Ta-da! Now this and all the information is given in this image. It's a time average map of NDVI. Here's my resolution monthly over July of 2020 in these regions in this scale. Okay, so you can also do the same thing. This was for the 2020. Now where's the 2021? Go to downloads. I'm just going to show you what I'm doing very, very quickly, why I'm doing this. Okay, so you can do side-by-side comparisons. Okay, so here's 2021 on the left and 2020 on the right. Okay, this is a very simplistic approach. It's meant for demonstration purposes. You can look at multiple seasons. You can look at this over the whole year. You can take your average over a chunk of years, say, or seasons over a chunk of years. Okay. Another thing you might be interested in, is this thing called KMZ. What happens if I just click KMZ on a downloaded KMZ file? Well, KMZ is a Google Earth file, right? It does this automatically for you. So I clicked on KMZ. I'm going to open the file. It's going to open it automatically for me in Google Earth. Okay. Now, say you're giving a presentation to some stakeholders, you had some other things you wanted to show, or more importantly, you wanted to display some geographic information over it. Well, I can zoom in on Google Earth. And now, look at this. You can have, I guess you can show your parishes, roads, okay? So you can really see some detail here. Now, I thought there was a way that you can show 3D mountains, some topography. I don't, let's see here. I'm not going to spend too much time on this because there's a way you can drape topography over this. I just don't remember how right now. But anyway, let's see, 3D buildings. There's trees. Nah, I don't think so. Anyway, I'm not going to waste my time on that. So yeah, so you can also look at your maps in Google Earth, okay? Now, these are just maps of single time periods, right? What if you want to look at the variability in that St. Elizabeth area over many years? Well, here's what we can do. We can do a time series of NDVI over one averaged location and look at that. Okay, so let's do this. So back to data selection. Okay, keeping everything the same, we're still looking at NDVI, but instead of a time average map, let's look at a time series area averaged. Okay, right here. Time series, what does that mean? That's just a plot, a line plot, like a stock market plot over a designated time, representing an area that you identify or you determine. Okay, so we're going to do a time series area average. Now, because I want to look at just the St. Elizabeth area, rather than looking at all of Jamaica, I'm going to zoom in a little bit. Get my hand there so I can move it over. I'm going to zoom in just a little bit more. Okay. And now let's just look at this. Oh, let me zoom it over a little bit more maybe. Yeah, it's right here. Okay. So now I'm just going to select this area right here. All right. So now that's our area of interest. And the time series area average is going to look at variability in this area. Okay. Now, let's look at the variability starting at, just to make it shorter because of time constraints with the Zoom call and data crunching. Let's just go 2015. Let's go January of 2015. And let's go, how about to October of 2021, okay? So now we've adjusted the dates to look at a time series of this area average, plot data. I hope it doesn't take too long. I mean, at most it would take 2 minutes, but we don't want to be sitting here on the call for 2 minutes. Okay, good. So it's subsetting some stuff. It always tells you what it's doing too. So it's good for the morale and confidence, because sometimes you can push these buttons, you have no idea if it's working. So this actually tells you. So it's averaging. Good. That's what we want to do. So I'm expecting to see a time series plot. Boom. Perfect. Wasn't long at all. Okay, now we see some interesting things here. This is the area average NDVI time series for that southern St. Elizabeth area, approximate, okay? You can see there's a minimum every year. There's, you know, I wonder if I can change this x-axis. Let's see if I can change the x-axis. I can't. Okay. I want to cancel that. What I was hoping to do is get rid of this white spot here. I just wanted to end right here, but okay. All right, let's look at this for a second. Let's see, 2015, 16, 17, 18, 19, 20, yeah, 21. So we're just estimating. You could count, these are months, right? So this would be January, February, March, April, May, June, July of 2015, August. So each one of these marks is a month. If you know when we started, you know what month that is, right? That's why I wanted to get a little bit more detail here, but it's not allowing us to do that. So look at this. We have a feeling that NDVI has a minimum, or I should say a total minimum, but a midsummer minimum in the southern St. Elizabeth area. The annual minimum is probably in the early winter months, right? The dry season. So let's say that this is July, am I right? January, February, March, April, May, June, July. So this is July right here. So here you have your maximum after the early season rainfall, say in April, May, early June. And you can see how the NDVI really responded in 2017, 18, 19, 20. Now, look at 2015 and 16. Anybody remember what the what conditions we had in Jamaica in 2015 and 2016? Anybody remember back then what the conditions were like?
It's kind of difficult to say, but probably fairly strong drought period. I don't know. That's
right. That was when they called the Godzilla El Nino. We had this big El Nino 2015, 2016, and that caused widespread drought in Jamaica, all kinds of water rationings. So you can tell that the NDVI for the area on the average was lower in the drought versus when you didn't have the droughts, right? You can also get an indication of these midsummer periods. So there's midsummer period here. You'd have to really count. So that's why I don't like the way this is organized. But here's what you can do. You can go to download. And now you can either download this image, you can download the plot, or you can download CSV. Now I like this, right? Comma separated value of all that data. So if you wanted to read it into Excel, or if you wanted a table.
Hello?
Huh?
You hear me?
Yes. Do you have a question? Okay, I guess that was a mistake. So now if I open up that CSV, what do we get here? So let's do this. Let's do this. I'm going to open up Excel, and I'm just going to read in that CSV. And I'm going to show you what we get, just as a demonstration. Here's Excel. I want to read in some data, text, and that was my downloads that just came in. Here it is. I'm going to open that in Excel, push the import, and it's delimited with commas. I believe this is how you do it. Yep, finish. Okay. Now, this is lovely. Now you can see here are all the dates, these monthly dates, and here's the NDVI values. Cool, so you don't have to stare and count crosses. You just download that CSV, read it into Excel as your text file delimited with commas, and you have the entire record here, right? I could even, let's see here, have just 2 decimal marks here. I'm going to format the cell to make it easier. Numbers, 2 decimals, boom. And then I'm going to align them to the left, I hope. Can I do that? Do I know how to do that? Yeah, there we go. Now, these are all my NDVI values next to the month. Okay, so let's look at July of 2015 was 0.6. July of 2016 was 0.63. We're in strong droughts. July of 2017, 0.69. July of 18 was 0.63 also. And July, okay, so all these Julys are pretty low. So maybe, let's see where the minimum is. Oh, look at this, August, 0.59. August 0.7 for 2016. August about 0.7. Yeah, so there is some variability here. So you'd have to look at and see what month of interest or what season, like what you'd really want to do is take the average over maybe August and July. That average there is 0.6. And then now for 2021, the average is almost 0.7. So you can see the range is very small between these values, but it's meaningful because it is an area average, okay? Anyway, let's go back to NASA Giovanni. And let's see, here's my area. Yeah, we're gonna go back here, okay. So what we've done here is we've been able to look at the time series of an area average and look at the variability. Maybe your interest is not in a specific drought, but maybe it's the average conditions like of the year, of the early rainfall season, the late rainfall season, the dry season. If you wanted to see how vegetation responds to that, using a time series of NDVI would be a very good way to do that. You could also look at it, compare time series from different locations. Now, let's go back to this area here. This area looks nice and green, okay? Let's look at a time series of this area we'll compare it to the time series of this of the St. Elizabeth area. Okay, so we're going to go back to time series, back to data selection. We're going to change our area. Instead of having this St. Elizabeth area, and you can get as detailed as you want. Maybe you'd get different results if you just average this area down here. You know, you can fine tune it to your area of interest, okay? But let's just, for demonstration purposes, go up to this area. And now we're going to plot that data area time series, that same period. So I'm guessing it's going to be a little higher. And what you could do, you could get the results from both areas in Excel and plot them on the same map. You could take those values, those comma separated values, and just make one plot, time series plot in Excel. Okay. Ah, look at that. It's coming up. Big difference, right? Much bigger difference. Look at these. The peak values are about 0.88 relative to these. They don't even get to 0.8. And you can also see it's a lot more consistent. The lows are the lower values every year are pretty high. They're 0.82, but they're all pretty consistently within the same value, about the same for the upper. Now, here's what you want to do. And this will probably be the last thing I'll do before I open it with questions. If I really want to compare one area time series to another, we need to have similar y-axis values, right? So let's do this. Let's put, let's have the range be 0.55 to 0.9 on both of them, okay? 0.55 to 0.9 on both of them. Replot. Okay, and now here, 0.55 to 0.9. And now the y-axes are going to be the same, and I can have a much more accurate interpretation. Okay, so I'm gonna toggle between the two. Here's that upper area, here's St. Elizabeth. Same y-axis, same dates, right? Look at the big difference. So you can tell right away that the NDVI in that northern area is a lot less variable and it's much higher. The NDVI, the vegetation index in St. Elizabeth, is a lot more variable and it's lower. Okay, this is just a demonstration in Jamaica. The objective is to use your knowledge on rainfall variability, droughts, for example, and then to see if there's a connection with how vegetation responds. Now, you also got to be aware that some plots are managed, they're irrigated, right? That's going to be less affected by your natural variability, where other areas that are less managed, in wilderness areas, for example, you'll get a much greater understanding of how the climate impacts the vegetation there. And you can use that information to prepare for droughts. Now, say, for example, you can look in the time series, you can go back in history, you can say, oh, here's how a big drought affects the vegetation in this area. And we know that because we looked at the data. So if you anticipate a drought of a similar magnitude, you can begin to prepare how that might affect that area in terms of vegetation. Okay, so that's a good way to use this information. So I'd like to open it up for any questions. I know it's been somewhat technical, but I hope intuitive and easy to understand using a very simple and free web tool. I use it all the time. It's very handy and it's very fast. Okay, so are there any questions? Okay, I do not, let's see here. Let me open the chat. Maybe there's a, where's my chat? There it is. Okay, so I do not see any questions. I'd like to ask a question and feel free to give an honest response. A, do you feel that this is an easy and intuitive way to look at how vegetation responds to rainfall variability, or is it difficult or confusing? So that's the first question. Second question is, do you see yourself maybe accessing this to evaluate vegetation variability in certain areas? So is it easy or hard, and do you see yourself using it? I'm not going to be offended if you say it's hard or you're never going to use it. I'm just very curious. And if you want to type in the chat box, if you're more comfortable typing than speaking, you can do that as well. Anybody?
Hello, good
Everyone?
morning.
Okay,
Hi.
great.
Okay, good.
Yeah, Leticia from Belize here.
Oh, hey.
I see that the vegetation variability at some point to our institute, which is the Sugar Industry Research and Development Institute, might be able, or we see that there will be the need of using it. What comes to my mind is we are currently embarking a project for land, seeing the land degradation. So what I'm seeing is we can have a comparison what was there prior to the intervention. And what can help us is to see what is the current vegetation. So whenever we do an intervention, we will see what are the positive or what are the changes in terms of the vegetation in Belize. I was trying to follow you using the different apps that were the different sort of things that you were moving there, but since it's my first exposure, I got really confused. Perhaps not confused, but I need to practice more, explore the different information that is there available, so that I can say master it and use it according to my needs. So
Okay.
it's not complicated. I know it's doable. It's not rocket science what I'm seeing,
Yeah.
but it really needs
If I'm doing
practice.
it, it's not rocket science.
Yeah. Thank you.
Yeah, thank you for your input. And also I wanted to say, based on what you had said earlier, if you're going to do a kind of evaluation of the land degradation, what would be a very interesting application is that in Google Earth, you can actually get archived aerial images of your area. So say, for example, it was natural and it was fresh and untouched in, say, 2012. You can go back and get the aerial photography of natural landscape in 2012. And then now, say, for example, it's been built up, it's been, urbanized. You can now get the present imagery. You can have that as background comparisons. So you can show, here's what the NDVI looked like when it looked like this. And now there's no vegetation. The NDVI is very low and the image looks like this. So you can definitely couple that. You have some valuable information to make that point. But I'm glad you tuned in. I'm glad it's helpful. And maybe if you have time, you can go back and listen to it again. And also what I'm going to do, I'm going to type my name in the chat. Sorry, not my name, my email. And if you want to learn more about it, if you have questions, if you wanted to do this, but you forgot a few things, if you want to explore some more in-depth applications, email me at any time. I just sent it. Oh, I got a question here. It's a direct message, but I'm going to go ahead and read it aloud. Can I redo how I exported the information Excel file using the CVS download? Absolutely. Okay, absolutely. So let's, you're welcome. I'm glad, I see the message, Leticia, thank you. So let's do this very quickly again. So I've done my map, I've done my time series plot. I love it, right? But unlike an image, like my map, I can download Google Earth, I can download a picture, but with the time series, I can download this right here, or I can download the actual numbers that made this. And to do that, I click on downloads. And now I have 3 different options. NetCDF, we're not going to go into that. PNG, that would be just the picture of what we just saw before, which could be valuable, just the plot itself. Or CSV, comma separated value. Okay, so I'm going to click on this. I just left click on a mouse. It automatically downloads. I don't do anything. And I don't know about your computer, but I think most computers, it automatically downloads to the download folder. And I take a glance at the file. It's some nasty name, g4.area, doesn't matter. All I know is the CSV file that was just downloaded. That's all I need to know. So now I know the file's downloaded. I can go to Excel. Open a new sheet. Go to Excel. Okay, a brand new Excel page. And now what I want to do is open that data. And to do that, I go to the data tab up here. Let's see, a little bit of a delay. There we go, data tab. I'm going to click that. Okay. And now I go to the left, it says, get external data from text right here. From the text. So I'm going to click on that. And then Excel is going to ask me, where is that file? And I know it's my downloads. And downloads, date modified. Let's reshuffle this. There it is right here. Go back. I can convince myself there should be there it is, CSV file right here. I'm just going to click that. And I'm now going to do import. Bottom right-hand corner, import. Okay. I'm going to have delimited. That's fine. And I'm going to do next. And down here, because it's a CSV file, means comma separated value, it's delimited with the comma. I just keep the comma. Keep the tab and the comma, why not? Now the comma has been checked. I'm going to go to next and just finish. And then it tells me where I'm going to put it. I'm happy with that. Okay. And there I go. So there's a couple of things I did. See these values? It's like 0.8163957. I just want 2 decimal points. I don't need all these decimal points. So I'm going to select all of these by clicking the first one, hold down Shift, Control, and the down arrow. I select them all. And now I right click, go to Format Cells. Click on that, down to number, decimal places 2, and OK. And now I'm going to align these to the left just so I see more clearly with the date. So I keep that highlighted, go to the Home tab. All right, and now this align left. Little button right here. So line left, boom. Now everything is brought right next to the dates. Easy for me to see. I'm going to change them all red. Why not? Just to make them stand out. And there we go. So here we have all of our values, right? And if I wanted to, I'm not an Excel wizard, but I could click this, select all those values and make a plot. I think we go to Insert, Line Plot, 3D Line Plot, and there we go. And I could do the same thing. Actually, let's do this. I'm going to put both of them together. Copy this. I'm just going to move these over here. Okay, so here are my old ones. I'm going, let's see, how do I add to a existing plot? Again, it's embarrassing. I'm not an Excel wizard. Can I add? I know I can do it. Anyway, I'm not going to waste my time on Excel right now. I'm sure it's super simple. I've done it probably 100 times. It's just been a while, but I can overlay that same data on this one too.
Sorry, Teddy,
Okay.
your hand raised. That's Kenton.
Yeah. Okay. Raised hand.
Kenton, do you have a question or comment?
The question
Kenton? All right, Teddy, it seems as if there's no activity there.
Let's see here. Maybe I can. Yeah, I don't remember how to do this. Anyway. Okay. So I see a comment in the chat. It says here, okay, okay. Oh, good. The drone pilot, yes. Two missions where I got India, 18 acres land. Oh, great. You know, so I see this message. Yes, okay. Well, I'm glad you said this because I wanted to bring this up. I see this message about the drones. Good news is that the CIMH just procured a drone with an NDVI sensor. And they told me someone in Dominica has it as well. So we can actually measure the NDVI given the spectral sensor on the drone itself. It measures the same things that satellites do, but higher resolution. So we'll be able to fly the drone over a plot of land and map that and compare the maps over time. We can do area averages, we can do statistics for everything we just did. You can do specific crops like, right, so we see here in Dominica, they're able to monitor crops such as tomatoes, carrots, peppers based on NDVI. This is great. And actually, I'm gonna write down your name because I'll probably be getting in touch with you to see how you're doing it, because I'll be probably managing the project in the beginning of the next year as we get our drone. So I'm very happy and excited to know that someone pretty much just a couple islands up I'm in Barbados, is doing the exact same thing. So I look forward to hopefully working with you and learning from you and comparing what we get. So see some other comments here. Something was not okay. Yeah.
Right,
so
that's Kenton back. That is back right now.
Is Macaulay the first name, or is Macaulay the last name?
Hello, Macaulay is the first name.
Okay, Macaulay, excellent. So I'm writing down your name, and hopefully I'll be in touch in early 2022.
Okay, no problem.
And I would love if you could send me your email in the chat. I'd love to email you and just actually have you send me some of your provisional results just to see how you guys are using it. I think that'd be very interesting if possible.
Yeah, no problem, no problem. I will.
All right, thank you so much.
Yeah.
Okay, so any other questions or comments? So yeah, I think the drone vegetation monitoring is going to be next level. I think we're going to get it's called precision agriculture, and what we'll be able to do is do precise monitoring on how vegetation responds to rainfall variability at different parts of the island where I live, but also we'll be able to validate drought forecasts. If a forecast says it's going to be drought, and we measure the NDVI and it's low or it's high, we'll be able to make any sort of corrections on the drought forecast as well. So a lot of valuable information to be used with the drone. I mean, 10 years ago, this was just a fantasy. You know, I remember being in school and a friend of mine had some interest in drones that just came out. And there was no such thing as a drone sensor or just like a camera. And he had this idea to put some sensor up there and monitor things. It was like science fiction. And now everyone can do that. I can buy one. So it's great technology. All right. Anyone else?
Yeah, there's still Kenton's hands that is
Oh, their
raised,
hands raised. Oh, good.
but
Okay.
not hearing from
Kenton.
him.
Okay.
There was a comment that his mic had stopped working. Kenton, can you hear us? If you have a question and there's an issue with your mic, could you maybe type it in the chat, please? So he does appear to have a question, Teddy.
Excuse me?
I think he has a question.
Oh, okay.
Just type a
wait for
question in
his
the chat, please, if your mic is not functional.
So another good thing about this, while we're waiting for Kenton, this is objective data, right? So we can actually apply numbers here, vegetation index values to different areas in response to rainfall, as opposed to just guessing or just like visually subjectively estimating. But always be aware there are limitations using, for example, the satellite data. It's area averaged. You have to know the area. If it's an urbanized area, don't fall in the mistake of saying it's vegetatively inactive because it's just buildings. Okay.
It's a quick question, Teddy, while wait on Kenton.
Hello, good morning.
Okay, there he is. Yes.
Yes, I just have one question. You said the resolution of the NDVI pixels is like 250 meters, if I'm correct?
That's correct.
So, and if I'm understanding correctly, the index itself, different plants will have a different index value.
Different plants. Well, different plants are going to reflect incoming sunlight based on their own internal characteristics. That's correct. And so that's the beauty of vegetation, is that each distinct species has its own characteristic wavelength reflectance, right? So it's going to depend on how that vegetation reflects the near infrared and the red. So even though the internal characteristics of the vegetation will be different, the ratio of the reflectance of these specific wavelengths will be the same.
All right, so in an area that they say like you have a mixed cultivation, how do you treat the data coming out of that area?
How do I treat the what? The data coming out of it?
If you have mixed cultivation, so it's not one crop, out of that pixel, how do you treat that data? You just, how do you recognize if that the value is saying that the crop is healthy, or, you know what I mean? So I'm trying to answer.
Yeah, so the value is just going to refer to the photosynthetic activity.
Yeah.
Okay, so the greenness. Now you can get very detailed, and you can actually look at specific wavelengths, but that would require a much different device, and it would be much more expensive. So, you know, to tell you the truth, I don't know if a tomato is going to show the same spectral reflectance as for these wavelengths as, say, like a carrot, but I do know the leaves on the tomato and the leaves on the carrots will be the same.
Okay.
Because they're all going to have the chlorophyll on it. But that's a good question to make that distinction.
Okay, thank you very much.
Yeah, you're welcome. Definitely. Okay.
Any other questions?
Hello.
Yes, go ahead, Macaulay.
Yeah, not a question, but probably just a comment to add to what was said last. Well, I guess using the NASA data, because of the resolution, it might be a little hard to determine the difference of crops or plants based on the resolution. What would be best is if you could use probably the same drone with an application like what we do, Drone Deploy. So Drone Deploy is a software that you use along the drone, and it gives you different photogrammetry output, such as NDVI and other things that you could open on QGIS, Google Earth, etc. So that would give you higher resolution outputs, and then you'd be able to monitor probably different type of plants or crops, etc. from that. But I don't think the NASA might be high enough resolution.
Yeah, Macaulay, your response makes me think that I maybe misinterpreted Kenton's question. So in the terms of the resolution, yeah. So the higher resolution with the drone will allow you to do things that Macaulay is talking about, is actually pinpoint specific areas in more detail, where the lower resolution of the satellite is going to have much less detail.
Just a quick question, Teddy. Could you potentially train train the data set to actually look at a certain, say, species of plant that may have a particular reflectance?
Yeah, you can, and that's actually called hyperspectral reflectance, right? So imagine any vegetation is going to reflect sunlight at many different wavelengths, right? But each species is going to have a predominant wavelength of reflectance. So a banana plant is going to reflect sunlight at a different wavelength, much higher than, say, a coconut tree, right? So what you would have to do is identify that peak spectral reflectance of each species. So for example, say banana, I'm just making these numbers up because I don't know, but say bananas have a very high reflectance in, say, like value X. You know what that X is, right? Then what you do with the hyperspectral sensor is you would just gather the data from that very specific wavelength. So each species, banana, coconuts, whatever, would have their own specific wavelength that you would monitor.
Okay.
But that's the basis of what's called hyperspectral reflectance. So you're looking at 255 different bands per species, where just regular reflectance or what's the word I'm looking, not hyperspectral, but multispectral, you're looking at like average of 10 bands per species. So the more detailed you get, the more information you can get. But just to put in perspective, when Macaulay and I buy these drones, we can get a multispectral or NDVI sensor for about $4,000 US. If you want to get a hyperspectral sensor, that's going to cost you about $60,000 US.
It's quite steep.
Yeah. And there's a lot of data that goes on with that too.
Okay.
Yeah.
All right, Teddy. Just want to thank you.
Oh, my pleasure. I'm glad we could spark some interest. And again, email me questions anytime. And Macaulay, I'll definitely be in touch with you, hopefully after the new year.
All right.
All right. Okay.
I think Dr. Allen has to depart now. We're going to break until about half the hour. By my time, that would be 10:30. Shall we resume then? There's a 10-minute break. At which point we will have a look at the soil physical properties aspects. Is that okay with everyone?
Yes, it is.
All right, so we'll resume at 1/2 the hour. Thank you. All right, so I think I'll probably start at this slide, just to look at, some of the services or ecosystem services that soils provide. Also to kind of look at, a physical breakdown of what soil is. So in this pie chart here, basically gives an example of the sort of physical makeup of soil. So mostly minerals, a little bit of organics, and then equal proportions of air and water. So they're generally like 3 phases of soils: the solid phase, there's a liquid phase. So this 50% here is a solid phase, the liquid phase, water, and then there's a gaseous phase, which is 25%. Now, soils actually provide numerous ecosystem services, to include the regulation of climate change, and of course, primarily production of food, provision of medicines. So, for example, some antibiotics and so on are actually have been discovered from soils. But perhaps 2 of the most critical functions is really the production of food, to feed growing populations, and then of course the regulation of climate, which it does through the presence of soil organic matter. All right, not touch on that too much since we're kind of mainly focusing on the physical aspects. All right. But in order to effect these functions, there's a combination of the physical, the chemical, and biological properties of soil that together affect all of these functions, the host of ecosystem services that soil provide. So here we just kind of want to focus more on some of the physical properties. And of course, just to bear in mind that these do not occur in isolation. All right. So the simplest of those is really looking at texture or the feel of the soil. So when you have a sample of soil between the fingers, how does it feel? Is it rough? Is it slightly grainy? Is it smooth, etc.? And the feel really is dependent on the presence of sand, silt, or clay within these soils. Just a sec, I see somebody's saying something in the chat here. Presentation mode. All right, just one sec. All right, thanks for that. Right, so if a soil is say dominated by sand, then the texture will be completely different, or at least a bit coarser than that, which is say dominated by clays, which are much finer particles. So it has a smoother feel. And by the way, if you have questions, please feel free to interrupt. I would much prefer if the questions come by way of voice rather than a text. It's a little bit more challenging to monitor the chat while doing the presentation, but you can interrupt me at any time. And in doing so, then everyone actually has, the opportunity to hear what is being asked. All right, so Sand particles are usually the larger, coarser particles, followed by silt, and of course, clay particles, which are usually about 0.2 microns or so, pretty smooth, right? But one of the emphasis we want to place here is actually on clays or clay particles. And this is so because clays actually impart perhaps the greatest amount of physical chemical properties to the soil, just by virtue of their physical and chemical makeup. Right, so clays are actually layered materials. You have what you call the 2-to-1 clays, and you have what you also call your 1-to-1 clays. And they're given this nomenclature because clays are made up of what you call a silicon tetrahedron and aluminum octahedral, which is actually pictured here. So this is how the tetrahedral looks like. So it's silicon with surrounded by 4 oxygen atoms. And then in case of aluminum, so it's aluminum surrounded by these hydroxyl atoms. Now, when you have one of this stacked on top of one of this, then that produces a 1-to-1 clay. So very good examples of that are what you call the kaolin groups of minerals. And a good example of that is kaolinite. In the case of the 2-to-1 clays, so you have 2 silicon tetrahedral sandwiching an octahedral, which is what is pictured here. And basically this forms 1 layer. So essentially you're looking at 2 layers of clay here. All right, so this is 1 layer stacked on another layer. And this is called a 2-to-1 clay, or what you call the expanding clay or swelling clay. So when these clays become wet, they actually expand. And when they dry out to a certain point, they shrink and they crack. Now, a part of this swelling property has to do with what you call these interlayers, which has the ability to take on water and some small molecules. So generally, this process is referred to as intercalation. All right, so water goes in here or in between the layers and swells the thing up. And that's not a feature that is common in 1-to-1 clays. So even when wet, they remain as regular soils. But 2-to-1 clays become plastic. All right, and here we just have a few, insets that are actually depicting some of the other aspects of clay. So for example, you're talking about the chemical properties that clay confer on soils. So clays go through what you call isomorphic substitution, right? So within these spaces, they are what you call base cations. So your magnesium, your calcium, your manganese. And sometimes what happens is that these cations can actually substitute for the main cations in the structure of the clay. So for example, aluminum, maybe, which is a 3+ cation, may be substituted by, say, magnesium, which is 2+. And that difference in charge creates a negative, well, it creates a charge on the surface of the clay, right? In some other cases, the aluminum cation in the octahedral can substitute for the silicon cation in the tetrahedral. Silicon being a 4+ ion, then that also creates a charge difference. And this allows the surface of the clay to be negatively charged. Now, by creating this negative charge, then this clay will now be able to, say, attach itself or interact with something of an opposite charge. So here in this micrograph, we're actually just showing what a pure clay mineral looks like in terms of being platy. So these little fragments here, which is a function, of course, because of the stacking of the tetrahedral and the octahedral. So these are just ways of studying clays. So one here we have an X-ray diffractogram, which basically tells here the main position of these two layers. Now, when the clay is intercalated, or when there is a molecule, whether water or some other small molecule between the layers, then the position of this band actually shifts. So this is a way of knowing, whether your clay might be occupied by something, whether it be water or some form of nutrients, etc. Technically, information of this nature might not necessarily be critical in agricultural aspects, but certainly in academics, because people use clay for a number of things. One of the most important property to actually, well, that clay actually confers on agriculture is what you call a cationic exchange capacity. So that's the ability of the clay, by virtue of the charges produced here, to actually attract other molecules and release them. So this is particularly critical for something like fertilizer application. So if you can tell the type of clay that you have in a soil, then that might help you with your fertilizer application. So your 2-to-1 clay has a different cationic exchange capacity than your 1-to-1 clay. So your rate of fertilizer application may be less for one clay compared to the other. So that is useful information that clays actually provide when you're looking at agricultural productivity. Now, the other thing to look at here is the way these minerals actually combine in soils. So sand is not just by itself, clay is not just by itself, silt is not just by itself, but they are in various proportions, and they combine to form what you call the texture of the soil. And that is represented here by what you call a textorial triangle. So it looks at percentage clay, percentage silt, and percentage sand. All right, so generally there are several divisions which are demarcated here with these thick lines. All right, and each of these give an indication of the proportion of clay or sand or silt that is present. So for example, if you look at this composition here, 40% sand, 45 silt, and 15 clay. So you basically locate the percentage clay on this axis, you go across, then the percentage silt, which would be 45, and you come down, and then the percentage sand, which is 40, and you go up. Now that gives a rough indication of the type of soil. So in this case, you end up with a loamy soil. One of the important things to note here is that of these three particle sizes, it takes a relatively small amount of clay to confer clay properties to a soil. So if you look at the loam, even though the loam is somewhere here in the middle, the percentage of clay in there is relatively small. All right, so that's crucial to note. Small amount of clay confers clay property on the soil. And of course, this has important implications for the way the soil is managed. So how you till, when you till, the amount of irrigation water. It will also impact things like your root penetration and so on. So information on say the type of clay is also important, because if you have a 2-to-1 clay, then you necessarily wouldn't want to work or to till that soil within the rainy period or in the extreme dry period. Any questions, comments? Anything you'd like me to clarify here? All right, I'll move on then. So again, just kind of looking at clay structure, you can see this sort of stacking in the clay. This, for example, is a 1-to-1 clay. Right, so this leads me to this figure here, which basically looks at soil structure, which is basically the spatial orientation or organization of the different sized particles within the soil. So it can be, say, for example, in a granular format, or like a prismatic format, or a blocky format, or a plate-like format, which, for example, the clays clearly highlight. And in the production of these different structures, of course, we have the involvement of some biotic factor, meaning exudates from plant roots, or we have exudates from microbial biomass. So it's almost like you're preparing mortar. You have your sand and you have your gravel, and then to that you'll add your cement and your water as the sort of binding agent. So that's the function essentially of the plant root exudates and the microbial exudates. And these exudates are generally, say, rich in amino acids and low molecular weight organic acids, and usually are highly nutritious. So you'll find, for example, and I'm not sure how many of you might be familiar with the term rhizosphere, where you're looking at the rhizospheric region of the soil, which is basically where the plant roots concentrate. You'll find that these areas are more nutrient rich and more microbial rich. And a part of the reason for that is the exudates that are produced by the plant roots. So microbes tend to feed on these significantly, and also produce their own exudates, making this area, particularly nutrient rich. So this is an area of the soil that basically encourages production because of its nutrient status, provided, of course, by plant roots and by microbial biomass. And in these micrographs, we're just basically showing the interaction of a pure clay mineral with some microbial biomass. So here you can clearly see the stacking, but once you've had the microbial biomass generating what you call extrapolymeric substances, then that is no longer visible. So it now appears as if you have, something that has been cemented over. So that's the nature of creating structures in soils. And again, the nature of this is really dominated are dependent on the dominant particles that are found within, each of these. So another important soil physical property we want to look at, or we had looked at, is the bulk density. And bulk density really is important from a management standpoint. So information from bulk density can, be used in, say, for example, the type of crop you cultivate in an area. It might give also, some insights into your water application, your irrigation water application, as well as how you go about employing particular soil husbandry practices. So when we talk about bulk density, remember in the earlier slide, we said that soil has 3 phases: your solid, your liquid, and your gaseous phases. Here, each is actually identified by a different color. So your solid phase, which is purple, if I'm seeing that correctly. Your gaseous phase is yellow, and your water, blue. So those are the main components of the soil. But when you're looking at bulk density, you're looking at all 3 phases, essentially, initially. By removing water, then you're left with the mass of the dried soil with air particles or air pockets, right? And a mathematical definition for density is the mass of the thing over the volume of the thing. Now, the volume of the thing you can find by various methods. So if it's circular, it's the area of that circle times the height of the thing. Well, cylindrical, I should say. All right, sorry, I see another chat here. Let's let me get this. All right, just 1 second there. I think I might have to let somebody in. Good, thanks for the notification. Right, so mass divided by volume. Now, generally, we use this bit of equipment here. It's called a soil corer to collect samples for bulk density, right? Which is a little cylinder stuck inside another cylinder. And then that is used to basically pick the soil up. All right, so determining the volume of that cylinder will be the area times the height, which is pi r squared h, right? Now the mass of the soil, the initial mass will be the soil in its native state. So that's water, air, and of course the solid components. And then that soil would be oven dried at 105 degrees over a period of time. Now this period of time is usually between 24 and 48 hours. Right? I generally go for about 48 hours because that allows you to dry the soil to a certain consistency, especially if these are clay soils. So because clay soils generally tend to have higher density, they're a little bit more difficult to dry. Additionally, they are able to hold water a little differently from, say, a loamy soil or a sandy soil. So I usually go for about 48 hours in the drying of these soils. So it's the dried mass at the end, the oven dried mass, divided by the volume that gives you the bulk density. Now in this chart here, it shows some nominal values for bulk density for different soils. And of course, it's important to note that the bulk density of a soil is a function of, you know, the physical properties of that soil. So whether you're sandy, silty, clay, right? So one of the things we find is that the high organic content soils usually have pretty low bulk density. So like your peat soils, which are generally organic rich, and so your compost, pretty low bulk density. But when you start looking at your clay soils, then the bulk density starts to get a little bit higher. All right. So how do you use bulk density in your everyday agriculture? Well, one, you can use it to monitor soil condition. So this will sort of inform you of things like compaction, right? And with the aid of a chart like this, or similar ones, you can also have information, or at least have an idea of what crops might be best suited to be planted in a particular area. Because compaction will affect bulk density, which will affect root penetration, which will also impact you know, your water holding capacity or water retention capacity of the soil. So if you have a soil with high clay content, bulk density will be high, but it might mean that root penetration will also be more difficult. It might hold more water, but less of that water might be available to the plants. So a simple bulk density test can tell you a lot about, you know, the type of management strategies to employ, or even the type of crops that might be suitable for this particular location. There's one other component here, which is particle density, which we do not really focus on that much because you know, it doesn't really provide a lot of information for agricultural purposes per se, but mostly engineering. And generally, your particle density would be about twice that of your bulk density, because by now you're actually removing your air component from the overall thing. Right. So that leads us to looking at soil water and looking at water characteristics and behavior in soil. Again, it's critical to remember that the physical property of the soil impacts the movement and retention of water within the soil. Right? But in addition to the physical property of the soil, we want to also look at the property of the water molecule itself, which is a standard thing. So water molecule is one atom oxygen covalently linked to two atoms of hydrogen. Now, in a covalent bond, essentially a pair of electrons is shared. One of these electrons is coming from the oxygen atom and the other from the hydrogen atom. And because of this type of bonding, there is generally no charge associated with that. However, each atom has what you call an electronegativity value. And the more electronegative that atom is, it's the more, so basically electronegativity refers to the propensity of that atom to draw electrons to itself. So the oxygen atom has a higher electronegativity value than that of hydrogen. So it has the propensity to draw these electrons to itself more. So the bond between, say, this oxygen atom and this hydrogen atom will mean that the electrons are not necessarily equally shared, but they are more drawn to the oxygen atom. Now, because electrons are negatively charged, this creates a partial negative charge on the oxygen atom and a partial positive charge on the hydrogen atom. If the things were equally shared, then there would be no partial charge. And that is what you refer to as a non-polar compound. In this case, the water molecule has what you call a polarity, again, because of the unequal sharing of the electrons. So you create a dipole, partial negative and a partial positive here. Now, how is this important? So this is important, for example, in the application of your nutrients, your fertilizers, because these usually come as charged particles, or at least when they dissolve in water, you know, that interaction, by this creating this partial charge, you have, you know, these, so your nitrates, for example, interacting with your water molecules because your nitrates have a negative charge on them, or your phosphates have a negative charge on them. And so those interactions actually provide the nutrients to the plants. Okay. Water molecules are also capable of forming what you call hydrogen bonds. So these are weak bonds between your hydrogen atom and your oxygen atom for two different molecules. Now, this sort of bonding creates what you call cohesion, right? Which is what is being exemplified here. So because the hydrogen bonds are weak, these bonds are easily broken. So if you pull the fingers apart, then that's that. But once the molecules are close enough, then they will tend to stick to each other. And that's cohesion. Now, cohesion is important because it allows or it facilitates water movements. The other force that is at play here is adhesion or adsorption. And that again is a function of hydrogen bonding, or it could be facilitated by these partial negative and positive charges that are created here. So in the case of adhesion, that is water binding to the surface of the soil particles. And again, you will have charges in your soil particles, or within that solid phase, you will have some charges, okay? The other force at play is what you call surface tension, which is basically speaks to the difference between the affinity of water molecules for each other relative to the affinity of water molecule for the atmosphere. So in this case here, where you see this sort of stretched membrane look over the soil, is really a function of your surface tension. Now, when all of these forces combine, you know, this allows water to move within the soil. Now, the movement of water through the soil column, you know, is termed capillary water. And again, is a function of the physical property of the soil. So if you look at your coarse sand here, bigger particles with bigger pore space, then the movement of water up this column is a little bit retarded compared to your fine sand or a clay mineral, where the particles are much smaller, and then that creates a much more narrow bore for the water to move through, which is also depicted here. So what this is showing is that if you double the diameter of the bore, then you basically halve the movement of the water up the column. So in your sandy soil, you would expect that, you know, your capillary water would be a little bit more retarded than say in your clay soils. Any questions? Right. So the other thing we want to look at is some of the energy associated with the movement of water in soils, right? So there are three main forces at play. You call your matrix force, your osmotic force, and your gravimetric force, right? Or gravity, sorry. So the matrix force essentially is looking at the interaction of water with your particles. And remember from the earlier slide, we said that the interaction of water with particles is essentially facilitated by adhesion or adsorption. Right? So in a soil that is saturated, you can see here, you have a reasonably high amount of water adsorbed to a soil particle, right? And there are two depictions of this. There's this circular one and this cylindrical one. And in here, what this is showing basically is that your soil pores are essentially filled or inundated with water molecule, right? And at this, you say that the matrix potential is high. Medium matrix potential, there is a limited amount of water molecules around that soil particle, right? And you can see all that's also been depicted here and across here. So your macropores are more or less vacant, and most of this water would be, say, sitting in your micropores, and that's around the soil particles. And then you have a dry soil where the water film is even much narrower. And in this case, water is actually, in very, very small amount, like a micrometer films, which are basically situated in the corners of these pores. So generally, to obtain the pressure potential of moisture in your soil, you would apply something like a tensiometer, which is an easy to use instrument. So it's just a water-filled thing with a porous tip, and it gives you a reading that's inserted into the soil. Now, basically, depending on how wet or how dry the soil is, that will determine how much water is lost from the tensiometer. And based on the amount of water lost, that will provide a reading here. Now, this normally comes in values of kilopascals, which you can essentially translate to the wetness of the soil or the dryness of the soil. And we'll see that in a few slides down, where you have a range of values that you can say, okay, these values are synonymous with this type of soil when the water capacity is at, X value. But we'll see that, as I said, in a couple of slides down. We're looking at soil water characteristics and so on. So soil water characteristic chart here. Now again, these values here that we mentioned earlier, kilopascal. Now, for these different particle types, or you could look at them as different texture of soils, sandy, loamy, clay, you will see that for a particular pressure measurement, if you go pick any one of these and you go up, then the volumetric water content is different. So let's look at sand. If you take this blue line and you continue up, for sand, you're basically at 0.2 meter cube of water per meter cube of soil. If you go up, it's just above 4, about 4.5 for loam, and just about 6 for your clay soils. All right, so Again, it's important when you do these measurements, so even if you're in the field and you get a measurement from your tensiometer, it is important that you're able to match this reading to a soil type. Because otherwise, the value that you pick up here might be misinterpreted. So if you had this value for a soil that you thought was sand or that you thought was clay, then you realize that if that soil was in fact sand, then you would have had far less water in that soil than you would have in, say, the clay soil. So it's important to have information on soil type when you're actually doing these types of measurements. And measurements with tensiometers can be done on farm by the farmer or through an extension officer. Very little training is required in some of these things. You just need to be familiar with these pressure potential values and then have some knowledge of your soil type to see, what your water content is potentially like in these soils. And of course, this is useful when you're considering like your irrigation management or your irrigation strategies. Right, so that's one way. But generally, when you're looking at water content in soil, or at least soil water, there are two things that need to be considered. So one is the content, and one, you're looking at your pressure potentials, right? Now, there are different ways of measuring water content. You have some real-time ways, pictured here, and then you have the conventional way, which is essentially the gold standard of collecting or determining soil water content. And in fact, this method is actually used to calibrate these methods. Right? So two types of water content: gravimetric, which is a mass sort of dependent calculation, or your volumetric. So volume dependent calculation, as the thing suggests, right? But fortunately, one can be determined from the other. So your gravimetric water content would be mass of water over mass of soil. Now, to determine your mass of water, same core collected that you could use, say, for example, your bulk density determination. When you take the mass of that core in its ambient state, then you have both water and soil. And then once that is dried down, so once that is oven dried for a particular period of time, then the difference between the dry mass of the soil and the nominal mass, or the original mass of the thing, will give you the mass of the water, right? And the mass of that water over the mass of the soil gives you the volumetric water content. Now, the volumetric water content times the bulk density of the soil divided by the density of water, you can use that to determine your volumetric, right? The density of the soil, of course, we have already discussed how you arrive at that by determining bulk density, which is mass of soil over volume of soil. The density of water is standard. It's either 1 gram per centimeter cube, or you can scale that up accordingly, but that is standard. So that allows you to determine your volumetric water content, right? So either you choose to use gravimetric, which is mass dependent, or volumetric, which is volume dependent. But this information, together with your pressure potential, say generated from your tensiometer, can be useful in your irrigation practices. And then just to look at a few other terms, which are some descriptors in soil wetness. So when a soil is fully saturated, we say it's at its maximum retentive capacity, which is also pictured here. And essentially what this is showing is that the airspace within the soil have basically been taken over by water. So it's full inundation. So maximum retentive capacity. Now, after some time, some of this water will actually leave the soil under the influence of gravity, which is one of the forces that we spoke about earlier. Now, generally, the first to go is the water within the large pore spaces. So those are easily influenced. Water in the large pore spaces is usually easily influenced by gravity. So that's what you call here your gravitational water. So that's just water that is just flowing down. So you're moving from, say, surface to deeper soil levels. Now, one of the consequences of gravitational water is that it leaches most of your nutrients from up top and take it below. It also removes some of your agrochemicals from the surface and dumps it deeper below, and sometimes to as deep as your water table. Right? Now, when the soil is, say, at after most of this gravitational water has drained out, the soil is now at what you call your field capacity. Right, so essentially water are now in smaller pores, and your pressure potential value is around about 10 kilopascals here. Right? Now further draining, so if you had a nude soil, there's no vegetation. If you had a nude soil, water will continue to leave that soil through evaporation. So more water is being removed from your small pore spaces, and then it takes it down to a point that you call your wilting coefficient. If there's vegetation present here, you will also lose some water through evapotranspiration. But at the wilting point or the wilting coefficient, essentially what happens is that the plant is unable to exert enough force to remove water from the soil particles, and thus wilting sets in. Right? Additionally, the difference between your field capacity and your wilting coefficient is where you find your available water. Right? And you have the optimal zone, and you have slowly available water. You can see here. Now, drying will continue further, dependent on your conditions, and that takes you down to what you call your hygroscopic coefficient. Now, essentially, that water is unavailable to plant and is only present in very, very thin films around the soil particles. So totally unavailable to plants because plants cannot exert enough energy to remove this. Now, this is something that we often see in soils with high clay content. So even though clay soils have the potential to hold more water, a good bit of that water remains unavailable because it's hygroscopic. It's tightly bound to the clay. And sometimes it's between those layers that we spoke about earlier. So the water is basically intercalated, where even under oven drying conditions, sometimes it's difficult to remove all of that water completely. So that is how strongly bound the water is to clay particles. And of course, it's important to remember that this is predominantly in the case of your 2-to-1 clays. right? Your 1-to-1 clays will function a little bit differently, but your 2-to-1 clays for sure, your intercalation will occur and you will have this tightly bound water molecules, these tightly bound water molecules to the surface or inner surface of the minerals. And that prevents the thing from being accessible to plants. And here we're just kind of illustrating some of what we said earlier. So you're looking at field capacity for different soil types. So you see your sandy loam, your silt loam, and your clay. And in all cases, clay basically tend to have, well, in almost all the cases, you'd find higher values for your clay. So it has a higher field capacity. It also has a higher wilting coefficient because a lot of that water is unavailable, right? It has also a higher hygroscopic coefficient. And the area where it falls down, which is most important, is the plant available water. So the plant available water is smallest, or smaller than most, even though the original water content or the field capacity is much higher. All right, so bear that in mind. Again, showing the importance of soil physical properties to water retention and water movement. And these are things that must be considered when you're looking at your application of your irrigation water, especially if water efficiency is of high priority for you. And again, this is the same thing being illustrated here. Your clay, high water content here, but a lot of that is unavailable. Now, how can you improve situation like that? So you have a soil with high clay content. And one of the ways is to incorporate organic matter. And this could be through the use of, say, your composts. And by doing that, you actually improve the water holding, but also the field holding capacity of that, or the field capacity, right? So remember, the field capacity is the amount of available water. Now, organic matter basically does this in two ways. So there's a direct way and an indirect way. So one, organic matter itself has a very high water holding capacity, which will increase your field capacity, which is the direct way. And the indirect way is that the organic matter basically helps to modify the structure of the soil. And by doing that, you increase the water holding capacity and thus the field capacity of that soil. Any questions there before we jump into the next segment? So remember that this is supposed to be interactive,
Hello?
And I'm sure that I'm not that clear to everyone.
Hello?
Yes, go ahead, please.
Yeah, what I wanted to ask was with the relation to the water and the water particles and the soil particles within the soil itself. I do some soil testing using a peeling test kit.
Right.
A water soluble soil test kit.
Yes.
But the results for both the macro and micronutrients, they give them to me in milligrams per liter.
Right. So milligrams per liter is the same as parts per million.
Right.
Yes.
So there is supposed to, Dr. Casimir was telling me that there's a bulk density test would help me to better interact those results so that I could get a better breakdown as to how much nutrients is available within probably like a meter of soil and so forth.
Right, right. So one of the things is that when you do the bulk density, so we'll give you an idea of say, because it sort of looks at compaction, it will give you an idea of the potential depth that that water could reach. But not only that, because you can also determine volumetric water content from a bulk density sample, then it can also tell you the depth of that water within the soil column. All right, so if you take a bulk density sample, you take a core for a bulk density, you determine the bulk density, you determine the gravimetric water content, and then you use the bulk density value along with the gravimetric water content and the density of water, which is 1, then that will give you the volumetric water content. Now you can use that volumetric water content, and based on how the soil was collected, you can tell how far up in the column that water extended. So it requires a little bit of calculation, but yes, it can be done. And if you know how far up in the column that water extends, and you have an idea of root penetration, then you can also speak a little bit about how your plants are accessing nutrients, right? Bearing in mind that you might also need some information on soil type and so on. So again, if your soils had a particular clay content, then that you could use to speak to the potential cationic exchange capacity of that soil. And cationic exchange capacity is actually important for understanding and determining how much nutrients a plant will take up. Because what the cationic exchange capacity does is that it tells you that, okay, in this soil, or this particular soil type has the capacity to hold and exchange this amount of nutrients per unit, right? So there's a good bit of information that you can use to determine a lot from that.
Okay, thanks very much. And essentially, I guess what you said last there, the available amount of nutrients within the area, that is what we really want to determine and translate to the farmer, so they will know whether or not that they would need to add said nutrients or not.
Exactly. So, which is a very important point, because one of the things we often find is that, especially where fertilizer usage is concerned, farmers generally over-fertilize, which has several implications. So one, there's a financial implications to the farmer, because it might mean that you're applying way more than you need to. And if you're applying way more than you need to, then that's a cost. So that's a cost in both the input and probably the labor to apply the input. And secondly, then there's the environmental issue. So whatever is unused may end up in some waterway, right? So it could be through runoff and end up in your stream, or it could be through leaching and it ends up in your groundwater, right? So this type of information can be used to actually determine whatever. And of course, you would also need information on how the plant uses the thing. But in lieu of this, one suggestion sometimes would be, and I guess this is one of the reasons farmers sometimes do mixed cropping, where you have plants feeding at multiple levels. So if, say, something escapes from your A horizon, then a plant that feeds deeper down may pick that up. So in essence, as we have a saying here in Jamaica, we drop off a head, drop on a shoulder. What one plant or one species of plant might miss up here is picked up by someone else down there. And it's not necessarily wasted, but it's a better efficiency than if you had, say, a monocrop and you were applying the same amount, if you had the same application rate. But very good question. Anybody else? All right then, so we'll jump into to drainage.
Excuse me, just one more question before you go on.
Yeah, sure.
I heard you made mention to something about traces of magnesium being present in clay soils in heavier quantities or something like that.
Right, so magnesium is actually one of the substituted metals in clays. And we have seen here in clay soils where the buildup of magnesium is extremely high. So you can actually physically discern it. And just looking at the soil, you can actually see that buildup.
Okay, so a question, in the soil testing, if would I be able to determine whether it's a type 1 or type 2 clay based on the levels of magnesium within the soil test?
It's a little bit difficult with just that information, because there might be magnesium present in soils that are even low in clay content. And again, it depends on the origin of the soil. So say, for example, the primary mineral that was present. So that primary mineral would have weathered and created that situation. Also, it might also depend on the presence of residual stuff from fertilizer application. So by itself, it could probably give you some information, but not definitively. And that is why sometimes it's important to have several different parameters when you're looking at soils, because soils are extremely complex. And the interactions there are also extremely complex and sometimes difficult to understand. So where possible, you can have, several pieces of information, then yes. So you could, for example, supplement this piece of information with just a regular soil texture analysis. So, okay, you're doing a soil textural analysis on this particular soil, and you realize that the different components are in whatever proportions. Now, you could probably use that to conclude that, well, the presence of magnesium here might be due to the presence of high clay content. But if the clay content is reasonably low and does not necessarily infer clay content on that soil, then that will help you to exclude the presence of that magnesium as being clay related. There could be some other pedogenic factor that is influencing the presence of that magnesium, or it could be a man-made factor, i.e. the application of fertilizer. You're probably picking up some residues from past fertilizer application. So, which leads me to say too, it is also important to have, or it will be useful if you have information on the history of that soil. So it has been in cultivation for X number of years, X number with these crops and this type of fertilizer application, etc., etc. So it's a good bit of information sometimes. Well, the more information you have, the better your conclusions can be. Especially if, you're using things like kits and so on, because kits and so on they provide rapid information, but they're not necessarily the most robust. Anyone else?
Thanks much, Joe. Yeah, thanks.
No problem. All right, so we're moving to drainage then. Looking at the fate of water in soils. So we're saying that when rainfall in a watershed, right? So precipitation here is equal to your evapotranspiration your storage, your drainage, etc. So you have a shower, water either runs off or seeps in, right? So drainage can occur in two forms. So you have runoff or you have your percolation. Okay, you're also losing water through evapotranspiration. Now, the storage is generally, depending on the amount of rain, would go through several phases, as we indicated earlier, where you have your loss of gravitational water down to your field capacity, etc., etc. Right, and this happens based, well, how quickly this happens is based on the type of soil that you have again. But of course, for agricultural purposes, and especially due to the water intensive nature of agriculture, ideally what you want is to have good storage, which ensures, good field capacity. Okay. So here, what this is basically depicting is a situation where these sort of, let me call them micro ponds, are created on the surface, and that is used to sort of hold the water in. By virtue of looks, this seems like it's a clay soil, so this water could be sitting there for a while. But even if it wasn't, by creating these, you ensure that you allow the water to linger a little bit more than on the surface, basically discouraging runoff, because once it's gone laterally, it's usually gone. But when it goes vertically, percolation, then it might linger in the soil for a little bit, again, due to the interaction of water and soil particles. So this sort of thing is encouraged. And again, just looking at some other things that encourage water storage in soil, or the movement of water in soils. So vegetation is one of them. If you look here, where we have these arrows going almost laterally and vertically, where basically the size of the arrow gives an indication of the amount of water that is moving in that direction. So where you have, nudity, it's going a lot that way and a lot this way, but usually more this way, especially because of the presence of the slope. But once you have included some vegetation, then this becomes a lot smaller and more water is going down through here. And of course, the level of compaction and so on, information which you can get from bulk density will also determine how much water percolates into the soil. Essentially what is being depicted over here. Now, interestingly down here, what this is showing is the presence of soil fauna or soil fauna activities in increasing your water retention or the amount of water that is stored in soil. So you're looking at number of ants, and I suspect that this might either be regular ants or termites, and these will tunnel through the soils and create these channels, and that will allow water to percolate in. So a soil of this nature would essentially have a lower bulk density. That is, a soil that has been tunneled through will have a lower bulk density than its counterpart. So the reason for that is that these the fauna activity has modified the structure. So it's kind of the same way you would add organic matter to modify the structure. It's kind of the same way these activities modify the structure and allows the soil to hold more water. Right. Just kind of looking at some issues. So here, this speaks to a perched water table. And then you have the regional water table. Essentially, the perched water table is a function of having some impermeable layer. All right. This could be something like a solid block of clay or some bedrock that does not allow penetration of water, etc. And then you have this perched water table. And then you have your regional water table, which is a true water table. All right. So just showing importance of drainage here. If you look at a plant, especially in early stages of development, planted on soil with, say, a high water table or fairly saturated, then what that does is that it retards root development. And when that soil dries out, because of the retardation in the early developmental stages of the plant, when this water is no longer accessible, then the plant struggles because the roots have not developed sufficiently to feed a lot deeper. Right, so having the right moisture content in soil is critical, especially in the early developmental stage. And in this case, drainage might be necessary to facilitate good root development. So what you can see down here is that the soil has been artificially drained, and now that early root development is much more robust. So when the water level has naturally gone lower, then because the roots have developed sufficiently in the early stages, it can expand a little bit more to now access water that is deeper down. So again, another important consideration when you're putting in your crops. Watch your water content. If it is too high and you have no alternative in terms of area to cultivate, then you might need or you might want to consider some sort of form of artificial drainage. And here just looking at some examples of artificial drainage. So you have your open ditch, pretty self-explanatory, I think, which you would essentially have on some sort of a gradient to allow your water to flow under gravity. And then you have these more fancy drainage system, more or less like your French drain sort of, where you have your pipes and these holes in the pipe that allows water to drain. Critically though, it's important that the holes are actually facing down to prevent clogging, which is what is evident here. So fairly inexpensive system, but less efficient. And in the case of high water table, of course, you know, you'd have to dig below the water table to get this out, well below. This is just looking at some advantages and disadvantages, or some benefits versus some adverse effects. So I'll not necessarily read that. The manual was sent out. I don't know if everyone has received it. So all of this information is present there. And of course, there's a recording, well, which doesn't really cover this, but the presentations, I believe, were made available. So this is information you can just read easily. So the next thing to look at is irrigation. So we're almost now in an opposite situation from having too much water. We have too little water. So too little water in a water-dependent sector means, you know, some sort of intervention. And that intervention is usually in the form of irrigation. several types. Again, some are more efficient than others. So again, you're looking at some sort of surface irrigation here where a ditch is dug and it's inundated with water. And that water is taken from one end of the field to another, right? As I said, the efficiency here is lower because then you're losing water to evaporation. You're also losing water to percolation, right? But one of the problems that this creates is what we have pictured down here, where depending on your soil type, you might have water pooling at one end and not necessarily reaching the other end. So if you have a loamy soil, then you will have a lot of water pooling at the initial end, which is here. So where the water is introduced, because a lot of that water will actually go down pretty quickly. And even though you have your thing on a gradient, because of the nature of the soil, then little water reaches the other end, or it does so at a much slower rate. Now, the converse might be true if you have a clay soil, where because the particles are a lot smaller and water might not percolate that easily, then at this end where the water is introduced might end up being a little drier, especially if you have a decent gradient. Then the water will move faster over the surface than it will through the soil. So the ponding is basically experienced at the reverse end, right? And then in this form of surface irrigation, to minimize some of the water losses, they have introduced concrete. So you don't really have that percolation, but of course you still experience some evaporation. And then the water is siphoned off into the field. Now with some of the effects of climate change, and at least some of the projected or expected effects, we know that higher temperatures, longer drought periods are basically in the forecasts. The idea of high water efficiency, I guess, becomes extremely crucial. So one might want to consider other forms of irrigation. So your sprinkler irrigation, for example, still not highly effective, but much more effective than the previous two examples of the surface irrigation that were put forward. In terms of costs, your sprinkler irrigation might be a bit more expensive to set up, but it does provide a little higher efficiency. But there are some disadvantages still. Because of the way the water is distributed, then it does also suffer from evaporation losses. And it does so in two ways. So one, you will have evaporation through heat. And then because the water is coming out in some cases as a mist, you also have water losses generated through the wind or by the wind. Okay. One of the advantages though is that it makes provides relatively cooler conditions for the plants. And because it does that, then the plants are generally able to thrive well. Another disadvantage that's outlined here is that based on the way it's applied, you have to be certain that you're actually making overlapping passes with your sprinkler in order to have your field covered adequately. And of course, this is probably one of the most extensively used because it's somewhere in the mid-range in terms of efficiency and costs. Well, perhaps the most effective type is the drip or micro irrigation, where water is delivered directly where needed. So you can look at these two plants. It's probably about 1.5 feet apart. But even then you still have dry spaces here because water is actually delivered just at the root of the plant, so just where needed. When a drip irrigation system is combined with, say, a water monitoring system, like using your real-time soil moisture probes we had pictured earlier, then you can create very effective irrigation patterns for your plants. There is this company they call, I think, Semtech, which provide this we call dig and drop system. Where basically you punch a hole, you drop a sensor down there, and that sensor tells you how much water you have and to what depth. So if you are aware of, say, for example, the root penetration depth of your plant, then because the thing is real time, you apply just enough water within that area. So you're not really applying water below where the root is feeding. So that is water saved. And that's a savings there for the farmer as well. Okay. The disadvantage though of this method, even though it's highly efficient, is that if for some reason you were to lose irrigation water under very dry conditions, because the amount of water that is applied is so limited, then this soil quickly becomes dried out. And then that creates a problem for your cultivation. As I say again, about 1.5 feet between these two plants, and even with water being applied, you still have a dry space. Right? And generally, this water would normally spread out pretty well. So that's one of the major disadvantages of this type of system. Apologies for the dog barking in the background. That's my neighbor's dog, if you can hear it. Any questions? All right, I believe we're probably on the last component, which is basically looking at thermal properties of soil. So, again, one of the things that is normally overlooked, I think, in soil management is, transfer of heat in soils, or how important that is. As this statement reads, that soil aeration is as important for soil moisture, but a little bit more difficult, right? Soil temperature influences numerous soil-based processes. So most of the activities that take place in soils are enzymatically mediated. And if you have done any form of early biochemistry, you will remember that enzymes operate pretty much like anything else at any other sort of biological system at a temperature maximum, right? And so enzymes need a particular temperature to be most efficient. So even like your microbial decomposition of organics in soils is enzymatically mediated. So microbes will actually spit out some enzymes and they will degrade some organics. And that degraded organics helps to provide nutrients for your plants. All right, so the temperature which this occurs, if it's at its optimum, then it will be most efficient. And if temperatures are low, then the efficiency will also decline. And this here just basically shows you a number of processes and how they are impacted by temperature. But as I said, that crucially, it's a little bit more difficult to regulate soil temperature, or at least less attention has been paid to, I think, regulating soil temperature than, say, soil moisture. And again, this is a component that is a function of your soil type. Because the amount of heat or so that is transferred is a function of how compact your thing is or the moisture content of the soil, all of which are related to your soil physical properties. And again, that is just being depicted here. So in here we're talking about solar radiation, which is the way the soil is warmed. So solar radiation reaching the earth. So a lot of it goes back out into space, you call albedo, and some of it actually enters soil. So it's roughly about 10% enters the soil. Now, if you look here again, using size of arrows to indicate relative amount of that physical attribute, you see in a soil that is loose, that the amount of heat transfer is relatively small, yes? But as the soil becomes more compact, then the amount of heat transferred increases, which indicates that heat transfer in soil is a matter of conduction, right? And then if you add some moisture to this compacted soil, you realize that there is even more heat being transferred. Of course, water has what you call your latent heat capacity. So it can hold a lot of heat, or it takes a lot more heat to get it up to whatever point. But again, conduction, the important thing here. So there's greater contact between particles, etc. And that's now been saturated by water. So the heat transfer is a lot greater there. So while it's difficult to actually manage heat in soil, there actually are a few methods to do so. Well, one primarily, which is mulching, but the different forms of mulching. So you have your synthetic or your plastic mulch, and then you have your organic mulch. Now, these provide other roles as well. So like your weed control, your moisture retention, and so on. So by controlling moisture retention, you can actually control heat, right? So the plastic mulch, we see that is often used, and this can either be clear or black. But this generally is increased soil temperature. So for people in temperate zones, they normally use this to extend like the growing season. Again, you know, heat being or soil temperature being important, or the thermal properties of soil being important for soil functioning. I'm sorry. Right, so I thought I had a thing of the organic mulch. Organic mulch, on the other hand, which is usually of some biodegradable component, usually your grass or your straws or whatever the case is, provides, you know, real cool conditions for your plants. So in the case where this actually increases your soil temperature, then the organic mulch basically cools your soil down a lot quicker. And of course, the issue with having too high of a temperature on, say, soils that are deficient in water or moisture is that you have rapid evaporative losses, and then that has an impact on the plant's ability to either access or to take up the water that is present. So remember that the more you evaporate and you get closer down to that wilting coefficient, then the more the plant struggles to take that water up because it cannot provide enough energy to overcome that energy by which the water is held to the soil particle, which is the matrix potential. Right, so all things to take into consideration. And of course, the use of organic mulch, as indicated earlier, it helps to regulate water loss. So your soil stay moist for a longer time because there's no direct penetration of heat from the sun, so evaporative losses are kept to a minimum. And then the final thing I want to look at here is, wildfires on soil properties. So they show where you have burning wildfires, and basically these volatile organic compounds, they get condensed and they produce these sort of waxy layers, which will of course produce some negative effects in terms of your aeration, in terms of your drainage, in terms of your water movement and retention. So how the plant takes up water, how water below this layer is distributed within the soil, etc. So something to note, especially if you have, like us here in Jamaica, sometimes where people practice a lot of slash and burn, especially farmers who basically occupy some of the hillsides. This is usually for them an inexpensive way of land clearance. Usually comes with other consequences, especially when fires burn out of control. But just to note that, these types of activities can actually alter some of the physical attributes of the soil. And of course, there's no denying the impact on the chemical and biological aspects as well. All right, so I think that's the last slide.
Before you leave, what I would want to, I just am making a comment here because in Grenada here, a lot of the farmers think that when you're planting things like cucumbers and pumpkin, they have to burn. So they would basically clear an area and they have to burn before they plant. And you try to explain the repercussions of that, but because it's tradition,
Right.
most of them don't really listen because they think it works for them.
I'm actually happy that you mentioned that because there is actually one advantage to burning. I'm going to stop sharing my screen here. There's actually one advantage to burning. And sometimes even people within, say, the forestry sector do what you call controlled burning. And the reason behind that is to generate a lot of phosphorus. So when you burn, you create a lot of phosphorus and it encourages rapid growth. So there is a benefit to controlled burning. It's not encouraged because, of course, it destroys soil structure, etc., etc. But then there is that component. And because phosphorus, the application of phosphorus fertilizer is particularly tricky, especially because it's so heavily pH dependent. I guess for some people, doing this is a way of getting around that. Because if you apply phosphorus, let's say outside of a particular zone around a plant, then it essentially is immobilized. So that might be a little tricky one. But one of the things that I think, I have learned over the years is not to necessarily discount some of the, the traditional means, or what you call the indigenous knowledge. Because sometimes, even though there is no say, well-explained science behind some of these things. They actually do work. You know, so I guess it's a matter of trying to evaluate the risk versus the rewards, and at what scale you do some of these things, because scale is also important, you know, so there's something to take into consideration. But thanks for that.
What I usually do is to encourage, even though they practice that, try and do crop rotation, do the light tilling of the soil, so you know you would, you know, decrease the compaction eventually.
Right. So, but you see, that's the thing. You fix one problem and you have another. So in the context of using soil as one of the ways to combat climate change, tillage is something that is discouraged, right? And the reason for that is that once you've tilled the soil and you've broken up those aggregates, then a lot of the organics that would have been locked away or secluded from decomposers is now exposed. And when that organics become exposed and it decomposes, then what that does is that, or what you call the process, what you call mineralization, then it releases CO2 to the atmosphere. So I think the whole thing is really, you know, to practice the thing with balance. Because there are certain things that you will not necessarily be able to get rid of altogether. So if you're gonna be tilling, then you might want to do some sort of reduced tilling rather than, say, extensive tillage. And even something as simple as tillage has implications for things like weed control and so on. So I think it's about you know, moderation of the practices and combining practices. So it's kind of like say the advent of your integrated pest control, where you use a multiple of strategies to control your pests. It's the same thing with your soil husbandry practices. I think sometimes, you know, in order to be able to produce at a rate that you can sustain food requirements, you have to do certain things, but at the same time, you're very mindful that these practices, when done unsustainably, can have impacts on the very same thing that you're trying to achieve. So sustainability, I guess, is the way. Sustainable practices, I guess, the way is, you know, so try to do the thing in a balanced way. Anybody else? No? I'm still failing to believe it was all that clear. But if there are no further questions and people would, you know, like to reach out via email, I guess you all have my email address already. You can do that. Forgive me if I don't sometimes respond immediately because of work pressures, but you know, you can always send me an email. I'm guaranteed to respond, maybe not necessarily immediately, as I said, but I'm guaranteed to respond. If there are like small groups that would like clarification on certain things, we can probably arrange for some convenient time where, you know, we can set up a little session and have some further discussion on some other aspects. And it doesn't necessarily have to be about soil physical properties, but soils in general, because that's one of my areas of study or research. I did see a hand just now. It went. So again, remember, this is supposed to be interactive. So, and it's a makeup session. We thought there would have been more interactions. But I guess sometimes you need a little bit more time, even with a second take, to digest some of the things. Now that the manual has been sent around, I hope that you've received it. If you haven't, please shoot me an email. I have it sent to you. But now that the manual is available, people can go through, you may have questions, or even if it's something just specific to your particular situation in your country of origin, so you can always reach out and try to assist as best as I can. So one last chance for questions, comments, concerns. All right, then I guess we will end there. So I just want to thank you for joining for the makeup session. Hope it was useful. Dr. Allen presented earlier on some of the modeling aspects, and of course with the soil physical property aspects. And as I said, I'm at your disposal. The project has come to an end, but I'm still willing to assist wherever I can. So please feel free to reach out and continue to stay safe. All right, have a good one.
Thank you very much.
Thank you.
Presentation.